Well, thank you all for coming tonight. Many of you will have come to our previous lecture series through the Alan Wilson Center. And as many of you will know, the Alan Wilson Center is no more. At the end of 2015, we didn't receive funding. So the University of Otago was very kind to provide funding for the Alan Wilson at Otago research theme. So my name is Lisa Madison-Smith. I'm professor of biological anthropology here at Otago, and I'm the director of the Alan Wilson at Otago theme, which basically provides funding for us to continue the outreach activities and the public lectures that the Alan Wilson Center developed and were so popular for many, many years. So hopefully that's how most of you have heard about the talk tonight. But also we have been busy attending the GSA, or Genetic Society of Australasia, conference here for the last few days. So some of you may also be here because you are attending the conference. So welcome, everybody. It is my great pleasure to welcome our speaker tonight, Professor Tom Gilbert, who's based at the Natural History Museum of Denmark. And Tom really is one of the top people internationally in the world of ancient DNA. He did both his undergraduate and graduate work at Oxford, and now runs his lab out of the museum in Copenhagen at the university. And we were very lucky to be able to get him to come out for the conference and to give two public lectures, one in Auckland, and many of you may have heard the Kim Hill interview on Saturday morning, and then tonight's talk. So you're not here to listen to me, but you're here to listen to Tom. So I will just ask you to join me in welcoming Professor Tom Gilbert to talk tonight about me, my dog, and Maize.
Thank you, and thank you for coming. I wasn't actually originally anticipating having people staying from the conference onto this, but I guess it's so cold outside that you can't leave. So thank you for staying on. There is a small overlap. As you can see from the title, what I'm going to talk about is dogs, maize, and not actually myself. I love to talk about myself, as do we all, but about other humans. And in particular, what I was going to talk about is how we've evolved in the world of ancient DNA, and in fact in DNA genetics in general, to be able to do genome sequencing on a very large scale, and how this is actually allowing us to see things that we can't see normally. So I am based in Denmark. I am not Danish, not yet. I am British. I promised I will not talk about the lions, so I will not talk about the lions, but thank you for letting them win once. And what I will talk about is, I guess move on. Do you need some help?
Good.
We're all good.
How do I full screen it again?
It's full screen up there. Oh, there you go. All this attention. Okay, so what I actually work on, people often ask me, what do I work on? What do my group work on? Because to be honest, it's a very, very schizophrenic mix of things. And I had to actually think about this in preparation for this. And at its sort of core, what I'm interested in is basically biodiversity. This is a nice picture I stole from the internet. And I like this picture because I was searching for biodiversity, and I like the fact it's shown as a tree. And the reason why I like it shown as a tree is because the nice thing about trees is you can take your chainsaw to them and shape them. And this is actually what I'm very interested in right now. How do we shape biodiversity, and have we shaped it in the past? And there's really two ways we've shaped it that are of interest to my group right now. One of them is we've got rid of biodiversity. So here we have an Irish elk, the great now extinct elk in Europe. They're very common across Europe until about 10, 12,000 years ago, and it died out, and we were probably to blame. And on the other example, of course, is the moa that New Zealanders managed to get rid of. And it's a very interesting topic, how we get rid of these things, and there's a lot of research going on. In fact, a lot of research right here is on this in the context of birds. Now, the other way we shape biodiversity is, of course, we actually build it. People tend to forget that we don't just get rid of it, but we do create it. We're very, very good at creating diversity. So what we have here is maize, just different versions of maize, all domesticated from the same original stock, and we've shaped it in many different ways. And so really what we look at is how did we get rid of diversity and how do we build it up again? And I'm going to focus on this latter thing, domestication. How have we domesticated things? Again, I apologize to those that were here earlier, but we have to go through some of the basics again. Domestication research has been very, very central to a lot of people studying the past. We're very interested in questions such as, where did we domesticate things? So we know, for example, that maize is a modified form of a plant called Teosinte, but where did we first domesticate it? When did we do it? Is domestication a fairly recent thing? Is it an ancient thing? What might this tell us about the people doing it? How many times did we domesticate things? And I don't just mean how many independent species, but I mean, how hard is it to domesticate something? So if you consider the wolf, is domesticating a wolf into a dog difficult, that it would only happen once? If you've read Clan of the Cave Bear, you will find out that they did it once in that. But if, on the other hand, was it actually really straightforward? It seems kind of intuitive that you see a cute little baby wolf and you bring it home, and you might eat most of them, but you might keep a few of them and you might domesticate it. And there's a lot of controversy over this. We really don't know how frequently things were domesticated. Once we domesticated them, how did we distribute it around the planet? How did we move things? This is a really interesting question because, again, it tells you about human and their behavior in the past. And the other thing that people really like to argue about is, how did we improve it? So once we'd got the original form, how did we change it into the form it is today? How difficult was that? And how do we study that? Well, the standard way of studying this is simply to look at modern variation. So you can, for example, does this work if I do this? Yeah. So you can, for example, simply look at the modern version of the crop or the animal. You can look at various wild versions of it. So this is Teosinte that I'm pointing at now, the wild ancestor of maize. This is some kind of in-between thing. And you can just look at it, and there's some very obvious things that stick out. For example, modern-day maize has got lots of kernels on. It makes it more enjoyable to eat. And so just, visual observations can tell you a lot about the present, but what it can't tell you about is what happened originally to get us there. Now, the way to start looking at that is archaeology. So, for example, people have been having a lot of arguments about domestication of dogs from wolves. And I said we don't really know when it happened, but there's arguments saying it was quite recent, arguments saying it was a long time ago. One of the reasons for arguing it was very ancient is because of archaeological evidence. So people have found skeletons of animals in Belgium about 32,000 years ago at the Goyer Caves, which are some very famous painted hunter-gatherer caves. And when they've been looking at these, they will see that some of them, for example, look very wolf-like, like the ones on, what side is that for you? Your left. And then the ones on the right look a bit different. And if you know your dogs, you'll know that one of the things that happens in domestication is the face gets shortened, the snout gets shortened. And when they find these 30,000-year-old skulls with a shortened snout, they say, well, we think this means it's probably a dog, therefore domestication is that old. Now, once DNA was sort of known about and people started to work with it and they could sequence it, we could use DNA to add to this. Now, of course, without DNA, you're constrained to things you can see. The beauty of DNA is it tells you what's going on elsewhere in the process. So the idea is you can simply extract DNA from the wild relative of what you want, of the modern domesticate, and you can sequence bits of the DNA and you can look for the differences between them and you can find the genetic basis of the differences. And people are doing this all the time on dogs and wolves. And for example, with dogs, one gene that we know is very different is one called AMI2B, and it just allows dogs to eat a more starch-rich diet, something that wolves wouldn't normally be eating. So again, we can spot that. And this really took off in the mid-2000s when we went through what's called the genomic revolution. A series of machines were built and they were sold in the mid-2000s that made sequencing an awful lot easier and allowed us to start sequencing whole genomes. So instead of looking at a gene for one particular trait, we could look at the total difference. And this is the area of genomics. And genomics is actually very hard to define what it is, but we tend to view it as sequencing DNA from the whole way across the genome, getting as much information as you can. Now, I'm going to take a brief detour here and tell you how I got into genomics. My background is actually in fish behavior. When I was an undergraduate at Oxford, the only thing I wanted to do was work with fish, and in particular, amphibious fish, the kind of fish that will go out of water and then go back in again. And I actually did a series of, in retrospect, very cruel experiments where I would take fish that are not at all adapted to being out of water, put them out of water, and sit on a stepladder and watch what they did. And it was interesting, but I didn't stay there for long. And I then moved in my PhD into doing genetic work. And I was doing standard genetics as it was done 15 years ago with the technique of PCR. We would analyze very short fragments of the genome, and it was very informative. But we then had this genomics revolution, and I thought to myself, I need to get in on this genomics, as did many of us. And so, and this is a true story, in about 2008, I was in Madrid, in this square, having a coffee. And I was doing what many scientists find themselves doing much of the time, if not all the time these days. I was sitting thinking, how do I write a grant to get me a load of money, to allow me to keep being fed, to hire people, to not get fired? It's a very positive world, academia. And as I was sitting there thinking, the problem is you need to find a new idea, and it's quite hard to find new ideas that somebody will fund. And I was sitting there and I was thinking, what can I do? What can I do? And true story, as I was sitting there, a pigeon crapped on me. Literally sitting there, splat on the head. And at first I was like, oh my God, pigeons. And then I was like, oh, pigeons. Because what I realized was my rage and hatred for the pigeon meant it might be a perfect study system, because what we biologists have a lot of problems doing is getting permission to sample things. It's really hard to get a bit of kiwi and sequence it. And I was thinking, everyone hates pigeons. How hard can it be? And they're actually very, very interesting animal. Pigeons, they're found everywhere. You can do a project that allows you to travel. As I said, nobody cares if you sample them. You can get permission to do it fairly easily. They've got a long history of association with humans. I love this because it shows a pigeon crapping on somebody else in the past. And it shows Jesus had a disciple to catch the crap being put on his head. It's a very wonderful animal, the pigeon. And it's actually got a very, very long history with humans. It was probably the first domestic bird, maybe 6,000, 7,000 years ago. And if you like domestication, that makes it interesting. Nobody else works on it because everybody hates it, which means that nobody's going to be fighting with you. Many good things going. It's also fundamental, actually, to Darwin's thought process for, basic evolution. Darwin was a pigeon fancier. He would look at pigeon breeds and he would say, well, they're kind of all derived from one pigeon. These things are changing. Oh, I wonder if things change naturally. And in fact, part of his thoughts that gave rise to the origin of species came from pigeons. These are actually illustrations from one of his books. And in fact, pigeons, they're hated today, but they didn't used to be. So a very little known anecdote is that when Darwin had written The Origin of Species, his publisher, John Murray, was trying to basically get people to decide if they liked it, give feedback, peer review, basically. And one of the guys he sent The Origin of Species to was a guy called Elwin. And Elwin read it, and he wrote back and said, basically, you should drop all this evolution rubbish. Just focus on the pigeons, because everybody loves pigeons. Everyone should love pigeons. So, they've actually got a long, distinguished history in evolutionary biology, and I wanted to get in on the action. And again, part of the reason why back then I wanted to do it with genomics was that genomics is quite hot now. It was very, very, very hot back in 2008, 9, 10. And in fact, if you look at the history of genomics, what you will find is that until about 2013, if you sequence the genome of anything, it didn't matter how pathetic your story was, what organism it was, you would get a paper in Nature and Science. Getting a paper in Nature and Science, I guess, is like beating the British Lions at every single rugby match. It's like the top thing you can do. It's really, really very important for academics, and so there was a good reason to do this. Unfortunately, things didn't stay that way. So by 2014, genomes were not quite as cool, but they were still being published in journals like PNAS, very, very good journal. By the end of 2014, they were being published in PLOS One, which is an okay journal. And again, I want to take a slight detour to tell you what happened at the very, very beginning of 2015. A genome came out in a journal called De Novo, which is mainly famous for having published only one paper ever. And this, again, is a true story. They published the Bigfoot genome in De Novo. Somebody actually, true, you can look it up if you don't believe me, somebody actually sequenced what they claim was Bigfoot's genome, and they published it in a journal they actually created to publish it. Because they said it was unfair, the world was not accepting their story in any normal genome. And what I really love about this, focus very carefully on Bigfoot, right? It's remarkable when you think about it, Bigfoot is modeled on Clint Eastwood. So it's amazing, amazing. And it's out there, you can find it. So there you go. So genomics was very, very cool. It's kind of fun now. But there is a serious point here, actually. Genomics has become actually, it's gone from a very rare tool where you could get a paper in Nature and Science because it was so hard to do it, to a very common tool that people make up stuff about genomics. And this basically means today, actually, we can pick any system, no matter how unloved or crazy. We can get DNA from the organism, we can sequence it, and we can get papers out of it, and we can really study things in ways we could never study before. So again, I was working on pigeons. I basically found myself a pigeon. This is what's called a Danish tumbler. It's a very noble pigeon from Denmark. I slaughtered my noble pigeon. I sequenced the genome. And I actually used it for my first genomic study. And basically, I did something slightly obscure, but I looked at a trait called the head crest. If you look at those pigeons, you'll see they've all got feathers sticking up on the back of their head, except for the black one, which has no head left at all. It's a remarkable headless pigeon. And what we did by looking at the genomes of a lot of pigeons was we found the genetic reason for why they had a head crest. And it's really not that exciting. But the point for me was I was learning, wow, this stuff actually works. We can do stuff. And this then gets me back to what I'm talking about now, domestication. And if we think about what I was saying before, that we can use morphology to study it, we can use archaeology to study it, we can use modern DNA to study it, this is very useful. It will tell us things like, how do things differ today? How is a wolf and a dog different today? It will tell us about when things first appeared, like when do we first see the snout reduction in a dog? But it has to be a very obvious trait. If you care about, for example, when could dogs first eat starchy foods, you can't look at a skeleton to find that. And in fact, if you look at many of the questions that I said were like central to understanding domestication, they're actually hard to study based upon modern variation. So if you want to know where things came from, when, how many times, how they were moved around the world, and you compare things today, you have to assume that the distribution of things today is the same as what it was in the past. And for a very, very simple example, if you were to go out into New Zealand today and start collecting animals you find, for example, cats, rats, these things, rabbits, and you knew nothing else, you might think they were native to New Zealand because you find them there. But of course, by looking in the past, you know they were brought here and things have changed. So change is a very important thing you have to account for. And some things you care about, for example, what was the order of the changes? You know, what came first? Did the reduced snout come first, or the ability to digest starch? You can't understand that by looking at the present. To do that, you have to go into the past. You need to time travel, and this is the beauty of ancient DNA, this area that I have a background in and that many people have. You can use ancient DNA to study the past. Now, ancient DNA is a very popular topic. I mean, it's most famous probably for Jurassic Park, a story which is about as far away from ancient DNA as you can get. What's actually very, very relevant for my talk today is that ancient DNA was actually basically created by Alan Wilson, the guy that this seminar series is named after. And Wilson and Higuchi, they were in the US. They had the idea in the early 80s that this DNA stuff that we know we can get by blending up mice and frogs and stuff, I wonder if it survives back into the past. And nobody had really thought about this, that the DNA might be in it. So they went to a quagga. This is a quagga. It's a horse-like thing that died not too long ago, maybe 100 years ago. They got some skin, and they actually managed to extract DNA out of it. And this was revolutionary in the time. 84, nobody had even thought this was possible. They actually made a phylogeny, and again, for the scientists here, got a paper in Nature based upon about 100 bases of sequence and 4 taxa, showing the complete obvious, that a quagga is more like a zebra than a cow or a human. Good old days of Nature, 1984, if only it was so easy. But again, the point was they showed it could be done. And these days, ancient DNA, this is just a classic range of things people work on. We love our skeletons. We love fossilized plants. Mummies are great. I love this mummy mainly for the mustache this mummy has. This mummy's called Sylvester. He has a name. People like his mustache so much. And actually, even that brown thing, that's a fossilized poo or a semi-fossilized poo. This is the kind of stuff we live off every day. Nothing gets an ancient DNA person more excited than old poo samples. We're a very glamorous bunch. And ancient DNA, like every other area of biology, has done very well for itself with the advent of the sequencing revolution. So we have these new sequences. They allow us to do genomics. They, in fact, allow us to do what's called Paleo genomics. You can actually look it up, and there's a definition online I found. It's the study of genomes of extinct organisms. This is not strictly true. Because most of my group work on paleogenomics, I kind of asked them to define it, and the rough definition I got was basically, it's a definition of genomics where you spend an awful lot of money. You're very, very surprised by the fact that every time you think you know what's going on, you're completely wrong. And in fact, this is a very important thing. People give you very old samples. They say they're very old. You spend a shitload of money on it, and then you find out they're only 25, 50 years old anyway because the samples got mixed up. So it's a very disappointing field, but it's also a very exciting field. and it's taught us a lot. And what I'm now going to do is do what I promised to do at the start and talk about what is this telling us about domestication. And I'm going to focus mainly on my two favorite systems, Maize and dogs and wolves. And hopefully you'll be able to show how now we can do genomes from this material on a big scale. We can just see things that are not otherwise possible. So Maize is domesticated from something called Teosinte. Teosinte looks like that. It's obviously quite different looking to Maize. Some bits of it are recognizable, like it's got leaves. Apart from that, though, it's quite different. And from the modern data, just the fact that we can see what's out there today, modern genetics, we know that it came in central Mexico. So I hope you can see this. There's a region called the Balsas Valley in central Mexico. It's a very tropical environment, not at all like Dunedin. And it's a very warm region. Yes, it's warm, tropical, moist. Probably was domesticated about 8,000 years ago. It's a bit controversial. By 6,000 years ago, it had moved to South America, and by 4,000 years ago, it had been moved to North America. And this was actually a very, very fundamental step in Maize's evolution, because the difference between the American Southwest and the Balsas region is that, as I said, the Balsas region is hot and sticky. The American Southwest is much drier, it's higher altitude, and maize had to be adapted to grow in this very different climate. And that is why you can grow maize everywhere today, because it adapted to grow in this whole new climate. If it had never done that, we wouldn't be able to grow it everywhere today. We wouldn't be able to enjoy cornflakes all around the world. So that's what we know. So then where does the ancient DNA come in? Now, there's a guy called Bruce Smith at the Smithsonian who really is the god of plant domestication. He's an incredible, incredible man. He's got a huge knowledge of domestication, and more importantly for me, an immense large buckets full of ancient maize samples. These are pictures of some of his samples. On the right, there's basically a modern dried maize cob, and as you go to the left, you see maize as it looked through time. And the very last one there is about 5,500 years ago. As you can see, it's very, very different. And so what we did was we took this sample and we simply sequenced its genome using all these fancy technologies in the hope we could see what did maize look like genomically 5,000 years ago. How was it different to the Teosinte that it comes from? So what had happened in the first 3,000 years? How is it different to the maize today? So, what did it still have to do? And we sequenced it and we did some basic analyses. This is what we call a PCA. It's basically just a way to visualize genomic data. And it shows a purple cluster, which is basically Teosinte. It shows a cluster on the right, which is modern maize. And bang in the middle, where it's that little red dot, Tehuacan, is our ancient sample. So it's just sitting between them. It shows it's a transition thing. Don't worry too much about that. But what was important was, I say, we could look at the genome, and we could look in the genome for genes that we know are very important for maize today. And we could basically see, do these genes look like the maize version that we have today, or the Teosinte version, the ancestral thing? By 5,000 years ago, how far along the process had it come? And that's what this figure shows. We basically see at the bottom things like genes to do with when it flowers, genes to do with basically where the leaves are. It was already in the maize form. So these were traits that people selected on early. They obviously wanted to develop it in a certain way. Things at the top, things like the plant height, things to do with glucose metabolism, they hadn't really thought about yet. And this is telling us straight away that the early farmers were very, very concerned about growing, what it was actually doing, as opposed to what they were doing with it. And I'm going to come back to that in a second. So once we've got one sample anchored at one time point, we can start filling in the gaps through time. And so the next two things here are maize samples from about 3,000 years ago and about 1,500 years ago. And we can sequence them, and we can do it on a population scale, actually, and get a lot of it. And then we can start to say, well, look, these changes happen first, and then these, and then these. We can just order that domestication. And when you do this, you actually find some interesting things. So one of the earliest things that is selected on, therefore the most important things to the early farmers, is a trait called shattering. Shattering is basically a very fundamental thing that many crops do. If you're a mama crop growing baby crops on you, seeds, you want to spread them forth into the world so they grow into new plants. And one way of doing that is to basically go bang when your cob is right. And you basically fire the seeds all over the place, and they spread all around the place as opposed to falling at your feet. That's fantastic if you're a crop and you want to grow new crops. It really is a pain in the ass if you're a farmer, because the last thing you want to do is get to your field and find all the seeds on the floor because they went bang overnight. You want to stay on the plant. And what we can see is that very, very clearly at the earliest stages of domestication, what the farmers cared about was stopping this behavior. They were selecting on plants that did not go bang and send their seeds everywhere. This makes a lot of sense. Later, as we move forward, then we find adaptation to things like dehydration, to different heights. So we can see they're trying to grow maize in different regions. And so all the adaptation is on that. So they fixed the annoying exploding issue. Now let's try and grow it elsewhere. What was most surprising to us is what comes last. Only about 1,000 years ago do we find genes under selection for things like the maize properties that allow you to make tortillas. They kind of make the sticky property of the flour, the ability to make popcorn, the sweet flavor in sweet corn. So this is very recent. And this immediately makes you wonder, like, what the hell were they doing with their maize for 8,000 years? Like, it was, Teosinte is not a very enjoyable thing to eat. And yet, it took like 7,000 plus years to finally make this crop we enjoy eating today. So very, very weird, not what we would have expected, but it does suggest that initially they just wanted to have something that would grow, would give them something, and only when they'd finally cracked that could they start messing around and making it actually enjoyable. So the other thing we can do, though, is actually another quite fundamental question in domestication. Today, people have been very busy sequencing Teosinte from all over the place. So what this figure shows is in purple and yellow, just different regions where you find Teosinte, the wild relative, growing. And people have been busy sequencing modern Teosinte to try and look at the genetic variation in modern Teosinte. And this is a very useful resource for ancient DNA people like myself, because as maize was domesticated in the Tehuacan Valley, in the Balsas Valley near Tehuacan, and as it moved north up this pass to get out, it would have to go through all this Teosinte. And this actually sets up a very interesting thing that we can test about domestication. So there's two fundamental models to how we domesticate plants, we being early farmers. In one model, you've got a whole lot of this Teosinte plant growing around the place. One day you have this bright idea that maybe instead of just traveling across the valley to get it, I can grow it in front of my house, and you start growing it locally. That's a very easy start. Then what happens is maybe you're looking at it growing there and you've got this population in your garden and you think, well, okay, maybe I like this one. It was growing bigger or faster or something. And you keep it and you replant it the next year, right? You retain the ones you like. And then you keep doing this through time and you keep retaining the things that you like. And so you're selecting on this population for a particular trait. So that's one model. It's a very straightforward linear process. Now, the other one is actually more complex. It requires a much more advanced thought process. But you start the same way. You're growing your Teosinte in front of you. But then maybe you hear about in another valley, there's another Teosinte growing that maybe has something you like. Maybe it's prettier, or it doesn't shatter, or it's taller. And you think, I wonder what happens if I bring that over and I mix it with my first Teosinte. So you basically transfer in new genetic material, and you keep doing this. You keep incorporating more and more variation by crossing things. This is no different to how we breed dogs today, for example. People look at a Labrador and a Poodle and go, let's put together and make a Labradoodle. The question was, though, were they doing this 5, 6, 7, 8,000 years ago in Mexico? And the beauty of the fact that we have all this nice Teosinte that have been sequenced from all over the range means we can look as maize moves through time and location to see, is there new genetic material being incorporated from the wild Teosinte it passes? And we basically see this as the case. This second model explains what's going on. It's very, very clear that the farmers, through time, were continually bringing in new genetic variation, and often from very long distances away from where the samples come. So people were really moving things around, crossing them in, and doing something that we would view as a very modern practice today, back 5, 6, 7,000 years ago. Now, bear in mind, at this time, there was not even, this was pre-Aztec, it was pre-Olmec, it was pre the big civilizations in South America. There was never any wheel in South America. So it was basically just villages of people, but they were doing this very advanced behavior. And we can only really see this through the genetic information. So a quick summary about maize. It's had a good 8,000 years of improvement. Weirdly enough, only recently become enjoyable, as I would define it. Very advanced crop improvement behavior going on, all viewed through the DNA. So with that, I want to use my other example now, dogs, right? And dogs, again, are a very, very interesting domestic study. There's a lot of debate over what's going on here. Where did we domesticate it? Some people argue, actually, it's an East Asian thing, especially when they sequence what we call the Y chromosome. Others argue that it was a European thing, so very different parts of the world. Another team, led by a guy called Gregor Larsen, who's actually started sequencing ancient samples, is being very diplomatic, and he's saying it was both. We had like European ones and Asian ones, and then we mixed them together to be a happy family. When was it domesticated? I've already mentioned this. There's a lot of controversy. Some argue it's an old thing. 30,000 years ago, when we were hunter-gatherers, we would have domesticated the dog and somehow used it for helping hunting or whatever. The other school of people say it's a recent thing. It's only once we started to build villages and have animals and farms to protect that we would have domesticated it. So maybe it's only 10, 12,000 years ago. This is a very, very fundamental big split here in opinion. How, again, we study it? Well, the classic way to study it today is very similar to what I described. You will study a load of modern dogs. You will find yourself a load of modern wolves, who are the ancestors. And ideally, once you've sequenced enough DNA from them, you would find one particular wolf, for example, this one here, that looks closer to dogs than any other wolf. And if you find this one wolf, I don't know which one it is in this one, it's probably not an Indian wolf, but if you find one wolf which is closer to all the other dogs, then you might reason, well, maybe this was the ancestor of dogs, because that's why it's closer, because it's the closest looking one. And if it was the Indian one, then you could say maybe dogs were domesticated in India, because that's where we find this modern Indian wolf that looks like a dog. The problem is with dogs is that actually when you look at wolves today, there is no wolf that has ever been identified which looks more like a dog than any other one. It looks very much like either people have not looked at enough wolves or the ancestor of dogs has actually gone extinct. It was a different thing. It wasn't a modern wolf. And it doesn't matter how many modern wolves you look at, you will never be able to actually identify what's going on. So the way we've tried to address this is by simply getting us as many wolves as we can, live wolves, dead wolves, archaeological wolves, sequencing them and building a huge reference database of wolves through time and through space, and then compare it to the dogs and see what we see. And when you do this, you can build a phylogenetic tree, which is basically a gigantic family tree. And if you look at this tree, what's important is the blue things are all dogs. The black things are modern wolves. The little turquoise cluster is actually ancient wolves, in particular wolves that were about 15 to 30, 40,000 years ago from Siberia, so Eastern Siberian extinct wolves. And if you flip this and look in detail, you find some key things. Firstly, you find there's a lot of wolves that seem to be very, I hate to use this word, it's the wrong word to use, but they're primitive in the phylogeny. They're basal, they're from North America. So North America we can see is very, very important. My American colleagues love this. I found this by Googling the amazing Donald Trump making wolves great again. There's not really any relevance for domestication for this, but I just had to show this because it's just, somebody's put a lot of work into it. What we also see, though, in this basal cluster of North American wolves is that's also where our Pleistocene, so our ancient 20, 30,000-year-old wolves, they cluster nicely with the American ones. Even though they're not found in the Americas, these remains, they cluster genetically with them. So it suggests, again, North America was a very important place. What's cool about these ones is we think they were hypercarnivorous. So we think that the wolves, until about 15, 10,000 years ago, were actually different to the ones today. They were like the dire wolves in Game of Thrones. They were bigger. They specialized on hunting big things like mammoths, the kind of last thing you would imagine you would ever want to domesticate. They were a much bigger, more aggressive thing compared to the tame little wolves you find today. And again, because we have a huge data set of genetics, we can do something very similar to what we did with the maize and looking for is Teosinte mixed in with it. We can look at what dog and canid species are mixing with each other. When you look at North American wolves, they've got a lot of coyote DNA in them. That's not surprising. Coyotes live in North America. If you put a wolf near a coyote, they will eventually share genetic material. That's the polite way of saying have sex. If you look at the ones from Siberia from 15 to 30,000 years ago, they've also got coyote in them. It's kind of weird because coyotes don't live in Siberia, but it probably points to the fact that the Siberian extinct direwolf-like things, they're not direwolves in the biological sense, but they are in the Game of Thrones sense, were probably in the Americas where they met coyotes. And again, as I said, this is not really very surprising. You have to watch any dog for a few minutes. Dogs, canids will basically try their luck with anything. But this is actually very cool information for domestication for a reason I'd like to explain. So if we zoom right in on our 6 truly ancient wolves, we see they cluster in 2 groups. You've got this group below the red line, which are about 18,000 years or older. In fact, they're actually 25,000 years or older. And we've got 2 above the line, which are much younger. They are 13,000 years and they are 18,000 years old. They've all got coyote in them, but when we look at how much coyote and what else is in there, we find that the youngest 2 from 18,000 years ago and from 12,000, 13,000 years ago have also got dog inside them. Now, this is extremely exciting for those of us who care about when were dogs domesticated, because it goes back to this question of, were they domesticated recently, 10, 12,000 years ago, or were they domesticated a long time ago in the hunter-gatherer area? The only way you can get dog inside an 18,000-year-old wolf is through either time travel, doesn't happen right now as far as I know, or through the fact that dogs were around 18,000 years ago to have dog, Pleistocene, direwolf mixing, and therefore give them the DNA. So if dogs were doing this 18,000 years ago, they had to have been domesticated 18,000 years ago. We have cave dog. We don't have farmer dog. And this is actually, you can never tell this from modern DNA. It really does say that this whole idea that we domesticated dogs when we had villages to protect, when we had cattle to herd, is not true. It must have been in the hunter-gatherer time. And then you can basically start to think about, well, why were we doing it at this time? Were we doing it to guard caves, or were we doing it to help us hunt woolly rhinos? Or did they basically domesticate themselves and move in with us? And I actually personally think it's the latter case. So a quick summary of dogs. North America is a very important place. As I said, dogs will basically try their luck with anything. And domestication is definitely pre-farming. Now, what I want to do in the last bit of the talk is move on to another of my favorite things, and this will be the human, as represented by the famous Australian, Russell Crowe. I hope you'll notice that one. Can we apply this to humans? Because, we haven't just shaped things through time. We haven't just shaped dogs, haven't shaped maize, but we've also been shaped ourselves. So humans have been famously shaped by the environment. We originated in Africa. We left East Africa. East Africa looks like this today. Probably didn't then, but the point is it was nice and warm. There were different things around there. And we had to adapt to new climates as we moved. This is basically meant to be England, but it applies, I guess, to Dunedin as well. Darker weather, cold weather. This led to a lot of selection on us. We had to adapt to it. And you can compare modern Europeans today and modern Africans today and see the differences and see we have these genetic differences. For example, Africans have got genes that give them basically darker skin, and that protects them from the sun and so on. We have genes that allow us to drink milk, which I'll come to in a minute. And the point of this, though, is that it's as with the maize and with the dogs, the question is, to me, at what order did these changes come in? Which things happened first? Now, in addition to moving, we had to adapt to new foods. I've already given you maize. We didn't eat maize before maize was domesticated. We've had to adapt to this much starch in our diet. We had to adapt to sugar, like again, as I'm told, the famous Australian dish, the pavlova. I was told never to say that in New Zealand. I didn't quite get it. But I did find one that I think's got a kiwi on it, if you look carefully. And this adaptation has been very, very fundamental. So an adaptation happens on a very, very long scale. Like in this hundreds of thousands of years. It's also very, very recent. I would argue the All Blacks have adapted to crushing England every time they play rugby with them. It's probably a very, very recent adaptation. Now, of all the adaptations, the one which is probably most important in at least recent time is this last one, pathogens. When we started to settle down in cities, or towns at least, villages initially, we set the conditions for pathogenic epidemics on the scales that basically would never have happened before. Too many people in too close a space. The fact that humans, didn't have anywhere to store the meat, would probably get drunk and pee in the well. It just sets up nasty, nasty pathogen situation. And we know that humans have been subjected to very strong pressure from pathogens in the last at least 9,000 years. And what we wanted to do was apply some of these techniques we've applied to dogs and to wolves and to maize to humans and see, can we also structure change in humans through time? Is there a way we can test this? And we are testing it. The place we care about right now is a place called Trondheim. Trondheim is in Norway, halfway up the Norwegian coast. It is even more dark and miserable than Dunedin. It's amazing. After there, anywhere's nice. It's very nice in the summer for about a day or two. But it's historically a very important place. It's not the capital today, but it was the capital. This is St. Olaf. St. Olaf was one of those good old saints that you could become in the old days for dubious practices. So he famously slaughtered thousands of Estonians, and God was so grateful they made him a saint. So he is the patron saint of Norway, buried in Trondheim. Because he's buried there until the 14th century, Trondheim was the capital of Norway. It was a big pilgrimage destination. It was very important for the people in that part of the world. And Trondheim's got the other distinction that they really have got a very well-documented record of what happened in response to the big thing that made life hell for most of the last 1,000 years, the Black Death. So you probably all heard about the Black Death. Black Death famously killed at least 25% of Europe's population between about 1350 and 1650. Really horrendous thing caused, we know now, by the bacteria Yersinia pestis. And what's really nice about Trondheim now, again, really sucked in 1350, this is a population trajectory of Trondheim as estimated through archaeological evidence. And so you see that from about 1000 AD, when the city's founded, it's very important. So Olaf is buried there. The population increases pretty fast. And then in 1349, plague comes in, and then everybody dies. Like 70, 80% of the population probably died very quickly. This is probably simplistic. It wasn't this fast, but the numbers fit. That's a lot of people dying. If you think about 7 out of 10 people you know basically vanishing next year, that's what happened. For the next 300 or so years, the plague basically kept coming back in cycles like it did everywhere else. The population never really recovered until the plague vanishes in late 16th century, and then it takes off again, bam, and goes up and up and up. Now, this is fantastic because that central period where there's a big drop in the population, you can ask the question, why didn't everybody die, right? Was it just because some people were lucky? Did some people have a better economic status so they could get rid of the rats? Or was it genetic? Did maybe some people have a genetic mutations to give them resistance to the plague? And this is what we've tried to test using these techniques I'm talking about. So it's quite simple to do. You basically get hold of a load of skeletons from before the plague, from Trondheim. You get them from after the plague. Now, Trondheim, as I said, is a cold, horrible place, but it means skeletons preserved beautifully. They've got an amazing skeletal record, kept in a very, very freezing cold basement, I should point out, so it's still not fun to work on them. But once you've got them, before and after the plague, and you can sequence modern-day Trondheimers in their traditional dress there, and you can sequence their genomes, you can compare what's happening through time. So what did they look like genetically before the plague? What do they look like after the plague? And what do they look like today? And what has been changing? If something has changed, there's really two reasons for it. Either so many people died and so many migrants came in to replace them that will change what they look like, or many people died, but a proportion of them survived because they were genetically, in some way, able to survive the plague. And the way you compare this is quite simple. You just basically look at what your DNA looks like before the plague and after the plague, and you can basically plot it out. This is a complex-looking figure, but it just shows the human genome across the horizontal, and it just shows positions that are quite different between people before and after plague. And you can do it before the plague in the present, and you can do it after the plague in the present. And whenever you see these big peaks, like here, for example, or here, that suggests it's a particular part of the genome that really is very, very different as a result of this time effect. And you can then plot them out in different ways, and you can look for, are there any unusual things in there? And so in this plot here, just look at the red dots. What the red dots are bits of the genome that are very, very different now to actually both before and after the plague. So in the last 300 or so years, they've really, really changed. And you can actually zoom in on them. They've got little strange acronyms after them. These are gene names. And when you zoom in on these genes, you find they're actually related to very important, interesting things. They're related to things like bone growth, muscle growth, inflammation. And this is actually quite interesting because people now are a lot bigger than they used to be. People are, generally more muscular, stronger. And this suggests that genes related to that have been under very heavy selection in the last 300 or 200 years. Inflammation's kind of cool. There's been more and more pathogenic challenges coming in. But also one of the real problems today that I'm sure many of you are aware of is our problem with allergies. People have got massively overactive immune systems, and there's been a lot of selection on inflammation, which in particular is what makes your life hell when you've got hay fever. But this is all stuff that's just happened in the last 300 years. What happens when we look at what I wanted to look at, which is the plague doing anything? And that's what this plot shows. Anything up in this corner is things that, again, have changed since about 1600 and the present uniquely. And things down here are things that changed before and after that big epidemic. And again, you find genes, and you can look at them, and you can see what they do. And we find basically two groups of things. All these ones in the bottom corner are genes that we know are very important for the immune system. Immune system fights pathogens. They're changing as a result of the plague. It looks very, very much like there were certain people who were genetically resistant to the plague, and therefore it actually made a big, big difference what was going on. The ones at the top are actually linked a bit to smallpox, which itself is interesting. Smallpox has been a very big problem the last 400 or 500 years, and maybe there's been also genetic resistance to that. So I'm going to stop on that. The point was that we can now take these ancient DNA techniques at the genome level, study biodiversity in this way, start to see what's going on. It's a technique that's getting more and more important. The main limitations are they were once money, That's not a problem anymore, because it's getting cheaper and cheaper to do it. Another main limitation was just simply there was not enough people to do it, but actually this is becoming more and more routine. I personally get very irritated when people view ancient DNA as a discipline. It's not really, it's like a standard everyday tool, nearly. The limitation is actually samples, and this is actually a real problem today for geneticists, because historically with ancient DNA, what we've done is we've gone to people with their valuable samples and said, can we have a tiny bit of one sample? Actually, sorry, historically we would say, can we have the whole sample? That's what we need to use, and then we would use it all up. Then we got very good at making methods, very good at working on tiny bits of sample, and we would get sample from one sample, and it'd be good. But unfortunately today, because we're in the population era, it means we're actually back to the destructive phase, because we need to use samples from many, 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 many samples. And that's actually, there's a mindset thing there. People don't like to give up many, many samples, but we can actually do many, many more powerful things when we get to them. So I shall stop there. I will again say thank you very much to Lisa and Michelle in particular, who invited me. And also I was in Auckland, where Craig Miller hosted me. A lot of people put a lot of work into this, in particular guys in my lab, but we have a lot of collaborators. And the funders, of course. And thank you for doing this and not going to the bar.
Thank you very much, Tom, for a very inspiring and great talk. I enjoyed it very much, and I'm sure so will you have. We have time for questions, and I'm sure there'll be many questions that you would like to ask Tom.
As long as they're not about the potato blight, for reasons I won't go into.
Yeah.
Okay.
There was an article recently about Dutch people being very tall.
Yeah.
There was arguing a lot with sexual selection for Dutch women to prefer Dutch men being tall. And they were saying, well, comparison with Americans who used to be tall but aren't so much. All this sexual selection, could it just be the Dutch who had more exposure to black men?
Yeah, maybe. I mean, so the changes we're seeing to do with height and body and stuff are actually things that have been happening continually since 1000 AD to the present. So they weren't actually linked to plague. They just seem to be a continual trait under selection for 1,000 years. Genetics is extremely complex. So the one thing we're learning right now is that we don't understand anything about how things, we're quite good at finding things. We really suck at explaining them. There's been some really great talks here just the last few days showing, for example, things like tertiary structure of the genome have such huge effects. So, yeah, I wouldn't dare go on and say what these things are actually doing right now. We really don't know, and it's a huge problem. This is actually why, if you go back to when I was showing these slides of the history of genomics and how up to 2013 it was quite easy to publish a genome, the reason it's so hard, or at least hard to publish a genome in a top journal now, is simply because people have worked out that just seeing a correlation is not enough, right? And they want explanations now. And to get a paper in Nature or Science now on a genome, you generally have to be either very lucky and have a very sexy story, or you have to do exhaustive amounts of actual functional work and these kind of things to see what it's doing, right? They've gone beyond the era of just saying, well, you've got a name and it does something, and hand waving's enough. Unless it's Bigfoot. Then you'll never get it published. So.
Could you tell us what tissue is best for ancient DNA when you, for example, you're 600-year-old people?
Yeah, well, there are different answers to that. One answer is whatever the curator will let you get away with. Sometimes you're constrained by not being allowed anything else. Generally, I mean, if you've only got bone, you've only got bone, but what you can do is sort of maximize your chance of it working. So the reason ancient DNA has historically been a complete pain in the bum to work with is twofold. Firstly, the DNA just breaks down and goes away. Secondly, the DNA gets full of DNA from elsewhere, so microbes like to crawl inside skeletons and digest them. And this makes everything very, very inefficient. So if you can choose tissues that the DNA is either better preserved, or basically the DNA degradation is slower, or they're more resistant to microbes coming in, you basically just make your life easier. And so we know, for example, bone, the porosity of bone varies a lot. Very old people have very porous bone, young people have less porous bone. So you can, for example, it tends to work out that young people bone works better than old people bone because they're less spongy, less bacteria can get in there. We know that some bone is more dense than other bone, so it's very hot right now to take the petrous bone from that part of the skull. Generally involves a lot of damage on the skull, but it's very, very dense bone. It's very, very well preserved. I actually kind of made my name in DNA by proving that actually a really good source was hair. Hair is a very overlooked source of DNA, but it's really great because when hair is formed, the DNA breaks down at that process, but then stops breaking down afterwards, and it's very, very resistant to contamination. So it's a very efficient source of what's going on. But basically, the sad thing about ancient DNA is it's all difficult, and how difficult it is and how expensive. It's never that easy a situation unless you're really lucky and find something deep frozen, which happens occasionally. And it really is very, very hard in this part of the world, in the Polynesians. People like Lisa are really doing the absolute hardest job. So.
Yeah, thanks, very interesting indeed. Most of us probably like to think we're getting smarter with time. So do any of the genes you find changes in indicate changes in brain development? I mean, either in us or in the dogs, presumably not in the maize, but anything would go that way?
Yeah. So again, you don't need to look at ancient samples to look at that. When people compare the modern genome of a modern dog and a modern wolf, they find, yes, it's not my area, but they do find genes that are annotated to do with neurological system and that kind of stuff. That doesn't necessarily mean things are getting smarter. It probably has to do with behaviors changing, and behaviors change new things. I mean, you could argue that we might well be smarter than cavemen at using Facebook. But if it comes to fighting off saber-toothed tigers, it's going to be a different thing. So that's the real problem with smarter. It's a relative thing, and it's in what context. But yeah, I mean, there's a lot of evidence that, and when you compare even humans to Neanderthals, for example, there's a Neanderthal genome, or even going further back to chimpanzees, you see a lot of selection is on genes to do with something to do with the nervous system. But again, this is where it gets a bit hand-wavy. It's like, what does that something actually do? We're less good at saying.
Oh, thanks for the fascinating human story. I'm just a bit curious about the immune genes you showed in the differences. Are they actually just the gene frequency change, or is actually those genes actually presumably in the HLA region, I suppose?
Yeah, that's a very great question. I would love to answer it, but I basically did that on the airplane. It is a frequency change, right, of the allele, because that's what FST is measuring. So it's basically an allele that was very rare. It becomes very, very common in the population. What it actually does, we're still working out. So we're actually right now running the analysis to see if it changes, the function of anything. So I can't answer that right now, but it is what we're basically looking at. And that's just to see if it has a functional effect, right? But whether that change in the coding effect then has a functional effect is a whole other question. And that is the more difficult bit that has to be done through years of graduate students crying, kind of stuff. Graduate students will recognize that. Yeah.
Are there any other questions?
Will the Lions win? Yes.
One more question?
No.
Yeah, thanks for the talk. I understand a lot of rapid evolutionary changes relate to preserving juvenile traits into adulthood. Are there genetic markers relating to those?
I don't actually know the answer to that question. I imagine there probably are. But yeah, I'm afraid that's not my area. I mean, a lot of people talk about domestication being a retention of juvenile features, right? So they say that a lot of dog breeds kind of look puppy-ish, right? Again, it's a bit subjective, but the idea is that basically you're somehow repressing the full development into kind of the adult form. And that's not really very surprising when you're messing around with the genome, right? You just basically knock out one developmental process by changing some gene at something that stops some regulator, and that will happen. But it is a curious thing that, when you look across domestic animals, and it is a bit subjective to anthropomorphization and you assigning things, but they do seem to retain these babyish traits. Yeah.
Great. Well, if there are no further questions, then please join me in thanking Tom for a great talk.
Thanks.